All right, good morning. I'm going to go ahead and kick us off. Welcome everybody to Grand Rounds. As always, I will call your attention to the very distinguished speakers coming up. Bonnie Halpern-Felsher, I think, has been everywhere, if CNN or Good Morning America, or testifying in just about every state under the sun. So it's wonderful that she's going to come back. We've been telling you about her work in bits and pieces in various forms, including a recent town hall that was standing room only. But it'll be wonderful to get a much more scientific presentation of where we are, as well as the speaker you can see on November 8th and was around traumatic brain injury. And then finally, this quality improvement series is something new. Been doing this for about a year now with Lane Donnelly as the chief quality officer. So I think I really encourage people to attend as well. We're working on some technology to help people get credit for showing up for Grand Rounds. And hopefully you've been seeing this each week. Make a note. And then, as many of you know, Becky Blankenberger, as our Associate Chair for Education, is really a leader in different sort of educational programs, and this just tells you more about the Engage and Educational Scholarship Series. And our next presentation will be from the library and about doing literature searches. And then I want to make sure we bring to everyone's attention that we're getting close to the date for the annual Maternal Child Health and Research Institute Symposium. We now have over 600 members in MCHRI. This is a research institute that spans across the entire campus, touching really everything we think is relevant to maternal and child health. We're very fortunate that Diana Bianchi, the director at NICHD, is going to be our keynote speaker. But it's also a phenomenal day because all of the MCHRI-funded investigators who present, both some of our senior faculty as well as more junior, and it's just a wonderful showcase for everything that's happening in maternal and child health research. Certainly includes good representation from the Department of Pediatrics, but it's a nice opportunity to see what's happening across not just all the other departments in the School of Medicine, but some really exciting work with engineering and psychology and other departments across the university. And with that, I'm going to ask Steve to do the introduction.
So good morning, everyone. Those who don't know me, I'm Steve Alexander, and I'm Director of Pediatric Nephrology at Stanford. And I'm really delighted to introduce Friedhelm Hildebrandt for the third time in the last 2 days. Friedhelm is the William Harmon Professor of Pediatrics and Director of the Division of Pediatric Nephrology at Harvard and Boston Children's. He has spent Wednesday, Thursday, and now Friday morning with us and UCSF as a visiting professor in both institutions. And as I mentioned, we've worked him pretty hard. This will be his fourth talk in 2.5 days. Before he begins, though, just a few words about his background. He trained in Marburg, Heidelberg, United Kingdom, and then at Yale, and then spent 7 years as Director of Child Health Research at the University of Michigan. He was recruited as Division Chief at Boston Children's in 2013 by the then Chief of the Division, Bill Harmon, and this was the only sort of targeted recruitment I've ever seen, where he said, I want this guy. And Harvard said, well, okay. And then they went and got him. So, okay. So he is a winner of the American Society of Nephrology Homer Smith Award and the International Society of Nephrology Alfred Richards Awards. And those are the two highest awards that those two organizations can give. He's a member of the German National Academy of Sciences and the National Academy of Medicine in the US, and this is what really blows me away. He has more than 300, that's 300 peer-reviewed publications, which for a pediatric nephrologist, I think probably sets the record. Over 19 of these papers have appeared since March of this year. And Friedhelm, on a very long and windy drive to UCSF or to Burlingame for our combined dinner last night, Friedhelm gave me the secret of how he's done so much great work. He has 15 research fellows supported by his grants, working tirelessly in his laboratory all the time, rotating, never fewer. And this laboratory has done some amazing things at Harvard. It's focused on the identification and functional characterizations of single gene causes of chronic kidney diseases in children. And his group has identified over 50 novel kidney disease genes and counting, still going. And his talk today is really, has a long title, but what he's really talking about is introducing true precision medicine in nephrology. And so Friedhelm, thanks for coming. Welcome to Stanford. You've held up a lot better than I have over the last 3 days.
Thank you so much, Steve, for your very kind introduction. I'm very grateful to Mary and Steve for the invitation. I had a great 3 days discussing with my colleagues in pediatric nephrology the great work that is going on here. It's really been a pleasure, great pleasure. Thank you so much. Today, I would like to broaden the topic that's printed a little bit away from just steroid-resistant nephrotic syndrome to chronic kidney disease in children. And I would like to start by introducing to you this young boy who at the age of 3 developed periorbital edema and edema throughout the body, also ascites. And this was due to nephrotic syndrome, which turned out to be steroid resistant. And when a biopsy was performed as expected, it showed focal segmental glomerulosclerosis, where you can see the fibrous strands here replacing intact glomerular tissue. And as unfortunately is the case in FSGS, within 2 years, his kidney function deteriorated and he had to be entered into the chronic dialysis and transplantation program. So if we ask ourselves, why does he have to go through all this trouble? There is at least a molecular answer, if you so will, which is shown in the next slide. He has a point mutation in the potosin gene that exchanges in the nucleotide sequence, which is shown here from the patient compared to the normal sequence, that changes this thymidine to a cytosine and thereby changes the GTG codon to GCG. And that in turn now encodes for a valine in the amino acid sequence rather than an alanine. That changes the structure of the potosin protein and thereby leads to loss of function. And a situation like this, in my mind, is one of the strongest cause-effect relationships that we can see in clinical medicine, where a mutation in one base pair of the 3.3 billion base pairs that we have in our genome is sufficient in and of itself to cause disease, in this case, focal segmental glomerulosclerosis. But as you know, this is just characteristic for thousands of diseases that we call monogenic disorders or single gene disorders, or also Mendelian disorders because they're recessively or dominantly inherited. And as you know, they're defined by the fact that in a given patient, like this child, the disease is caused by a mutation in one gene only out of the more than 20,000 genes that we have in our genome. Now, a disease gene can act in a recessive way or in a dominant way. If it's a recessive gene, like here, then both copies that we have for most genes from our parents, one from mom, one from dad, need to be altered or mutated for the disease to occur. The parents will be healthy heterozygous carriers, for instance, such as in cystic fibrosis. And for most recessive disease genes, heterozygosity does not mean that individual will develop disease. So if you take a family history, it will be negative because at worst, any ancestor of the parents will also be a healthy heterozygous carrier. And in contrast, in dominant diseases, if your mutation exists in one of the two copies that we have for most genes, that is sufficient to cause disease. Therefore, one of the parents will have the disease, and in the parent's ancestry, the disease will have segregated throughout many generations. Now, the term monogenic does not exclude, however, that in different patients, different genes may cause a very similar disease. For instance, for steroid-resistant nephrotic syndrome, there are currently over 47 recessive and 8 dominant genes that cause steroid-resistant nephrotic syndrome. Now, if we ask what is so special about monogenic diseases, then let's look at the flow of genetic information from a gene to a disease phenotype, or in the normal case, just from a gene to a healthy phenotype. About 70 years ago, Francis Crick formulated the so-called central dogma of molecular biology, where a gene is expressed as a messenger RNA, it leaves the cell nucleus, and in the cytoplasm at the ribosome, it's translated into a protein, and the proteins together determine the phenotype of an individual. I don't know my Greek well, but I think proteo means something like I'm the boss, I'm the most important one. So proteins are very important for organismal function. They're enzymes, as you know, they're structural proteins. And for instance, if I look at my hand, I'm probably mostly looking at the keratin protein. There are also lipids and sugars. If you work on those, I can see that. But proteins are very important, and they're encoded by single genes. So if you have a single gene mutation, you actually have the primary cause of disease in hand, because the gene with a mutation will be transcribed into the messenger RNA that has a mutation now, and that will lead to a defective protein, which for instance could be truncated or shortened, thereby losing its function and thereby causing disease. So with the gene in hand, since the mutation represents the primary cause of disease or the etiology, you can now offer unequivocal molecular genetic diagnostics to the patient and the family. You have a secure starting point to starting this black box, which is the pathogenesis, which is still mostly unknown in nephrologic disorders. And then when you can start the flow of information, and in this case, the flow of misinformation, so to speak, then you gain novel insights into disease mechanisms, and sometimes you realize that there's new mechanisms of physiological cell function. Also, with a mutation in hand, you can often immediately draw genotype-phenotype correlation, where mild mutations may cause late onset of steroid-resistant nephrotic syndrome, whereas more severe mutations may cause early onset. And we hope that through better characterization of disease mechanisms by finding these single gene causes, that at some point we can target specific therapy and new therapy to patients with these disorders. So if we again think about disease causation in genetic disease causation, then we can consider a spectrum like this, where I drew a gradient from nature to nurture, or in other words, purely genetic disease causation to environmentally caused disease causation, from red to green. And if you think, and let's start at the blue end, trauma, we probably all agree, is completely external. However, a child with ectodermal dysplasia has a problem with heat sensation, so he or she is prone to burn trauma. So there could be actually a monogenic risk factor or component even to an external disease cause such as trauma. And even in toxic influences, which are mostly external, in glucose 6-phosphate dehydrogenase deficiency, the patients cannot have fava beans, otherwise they develop hemolytic anemia. So again, there can be a monogenic influence in rare cases. Infectious diseases are mostly external, you would agree, but there can be genetic disposition, the same degenerative diseases, which are often polygenic. And then in pediatrics, many developmental disorders are plainly monogenic with many different genes involved. Metabolic disorders, which our pediatric colleagues solved almost decades ago before the human genome was even known, are mostly monogenic. And for the two monogenic terms, I drew dominant before recessive, because in recessive diseases, you have full penetrance, whereas in dominant diseases, sometimes you can have incomplete penetrance. That means throughout a pedigree, where everybody, one individual in each generation has the mutation, one of these individuals may actually never live to see an expression of the disease. And that's an ill-understood phenomenon, which reduces the genotype-phenotype correlation, and that happens in dominant diseases and very, very rarely in recessive ones. To look at that in a more quantitative way, I drew a chart here where I assume we have 100 patients with a kidney disease, let's say steroid-resistant nephrotic syndrome, so that's the 100%. And we see how at what age they manifest between birth and 100 years old. Let's say there's a patient with an homozygous truncating mutation in potosym, in this autosomal recessive gene. And usually these age of onset curves are sigmoidal, so they have a characteristic where there's a median age of onset, which may be 8 years or so in this case. For many recessive disease genes, even with homozygous mutations in the same gene, homozygous meaning the identical mutation from mom and dad, you can have minor mutations that bend these curves over to the right with later onset of disease. And then as a tendency, dominant diseases manifest more likely in adulthood than recessive diseases. It's actually characteristic in pediatric nephrology where autosomal dominant polycystic kidney disease has adult onset mostly, and autosomal recessive polycystic kidney disease has child onset. So in a dominant disease genes, which is shown here in purple, there can be the phenomenon of incomplete penetrance where a person who carries the mutation does not live to see the disease manifest. And then again, different mutations in the very same autosomal dominant gene may lead to a milder form of disease with later onset. So Mendelian genetics, in a way, is very clear-cut once you accepted the concept that in a recessive disease, you have to have both copies of the gene mutated before you can accept that these mutations are causal. Then it's very clear-cut. However, there are a few genes where it depends on the specific mutation whether they behave in a recessive or dominant way. For instance, in renal stone disease, you see this sometimes that certain mutations need 2 mutations for renal stones to occur, and some other mutations behave in a heterozygous way, in an autosomal dominant way, and the disease segregates through multiple generations in a family. So Mother Nature, unfortunately, can make things worse by shifting these curves over to the left in way of gene-environment interactions of toxic substances. And we hope that we can reverse this by understanding these diseases and shifting some of these curves back over to the right with novel treatment and prophylaxis. So our world as pediatric nephrologists is kind of summarized in this slide. And Steve Alexander has been very active in the Nephrotic study over decades for the North American Pediatric Renal Transplant Cooperative Studies, which shows here in about 9,000 children and young adults with chronic kidney disease who manifest before the age of 25 years with their chronic kidney disease. And that's the cohort that I'll be keep talking about from now on. In this group of patients, 88% of everybody, so that's 100% minus these 12%, fall into one of these colored categories. Almost half of individuals who develop chronic kidney disease before age 25 have chronic kidney disease because they have a congenital anomaly of the kidneys and urinary tract. That's the acronym FOOD. 15% have steroid-resistant nephrotic syndrome, like the patient that I introduced to you. 14% have glomerulonephritis, and I'm including here, for the sake of simplicity, atypical HUS. 6% have cystic kidney diseases such as autosomal recessive or dominant polycystic kidney disease, forms of nephronophthisis, and so on. And 3% develop chronic kidney disease because they have nephrocalcinosis with stones. And about, say, 12 years ago or so, in pediatric nephrology, we wouldn't have expected that genes play a major role in these disease categories. However, in the meantime, there are over 240 genes known that in a monogenic way, mostly recessive, sometimes dominant, cause any of these forms of disease. So for Kaput, there's 45 genes right now. For nephrotic syndrome, 55, for glomerulonephritis, 10, you include atypical HUS. For cystic kidney disease is 100, and for nephrocalcinosis, 30. And we discovered, mostly by whole exome sequencing, 80 of them, and then when we find them, we study the cell biology, make animal models to understand what the pathomechanisms are. So when I was a fellow, I wrote a grant to find the gene for nephronophthisis. That was back in the last millennium. and took me 7 years to find it, not knowing that 30 years later there'll be 100 genes in this category. But there's a new gene pretty much every 6 to 8 weeks. So with these many monogenic causes of chronic kidney disease before age 25, you may ask, in how many of the patients can you actually discover the causative mutation? And that stands at about 20% right now altogether, where it's about 20% in acute, in that range in nephrotic syndrome, 14% here, but it's 70% in cystic kidney disease, and I'll show you later in echogenic kidneys as initial finding. And then again, about 20% in nephrolithiasis, nephrocalcinosis. But now let me go back for a moment to the child with steroid-resistant nephrotic syndrome. Here is where the disease happens. That's the renal glomerulus with its 3 layers. The endothelial cell layer that's fenestrated in yellow. It's wrapped into the basement membrane in green. And on top of that sits the epithelial cell layer, which are the podocytes in red. And the mesangial cells hold it all together. In 2 weeks, I'll go to the American Society of Nephrology meeting, probably for the 32nd time with no interruption. It's a major meeting. And I remember that for the first 20 years, there were hundreds of abstracts that tried to incriminate this little blue guy, the mesangial cell, in the pathogenesis of steroid-resistant nephrotic syndrome. And why was that? This was the only cell type that nephrologists could culture. So we were really looking for the car key under the streetlight. But when Corina Dignac found mutations in podocin, she realized it's not expressed in mesangial cells at all. It's only expressed in podocytes. That's why she called the protein podocin. When Carl Drückersen found nephrin, another monogenic form of steroid-resistant nephrotic syndrome, again, it was expressed in the podocyte. For the 55 genes that we know now, they're almost exclusively expressed in the podocyte, hence the term podocytopathy for steroid-resistant nephrotic syndrome. So the podocyte is a neuron-like cell. You can see it sit here like an octopus. It hugs the glomerular capillary and keeps it tight with its primary cellular processes, which are tubulin-based. It shoots off at 90 degrees actin-based secondary processes, and they shoot off tertiary ones, which are in these little holes here, and they interdigitate with neighboring podocytes, the yellow one with the blue one and with the red podocyte, to form the glomerular slit membrane, which is part of the glomerular filter to keep protein in the bloodstream. And in steroid-resistant nephrotic syndrome, this very intricate structure is effaced. So it's very plump, and thereby protein leaks out, and you have protein loss. And due to the decreased oncotic pressure, you have edema and ascites evolving. And then finally, fibrosis, because some of the podocytes drop off and are being replaced by fibroblasts. We are born with as many podocytes as we hopefully have at age 80, because these are permanent cells like neurons. That's why it's very important that podocyte function stays intact throughout life. And I'm giving you blow up now a little cut across the glomerular slit membrane, where you can see the slit membrane here. Down below is the endothelial capillary, with the glomerular capillary with the endothelial cells in yellow, wrapped around by the glomerular basement membrane. Here it's a big fat podocyte with a cell nucleus with a large foot process that's abutting a neighboring podocyte's foot process, and here forming the slit membrane. So blood will be filtered through the fenestrate, seeping through the glomerular membrane, then going out here into the Bowman's capsule as the ultrafiltrate in the filtering process. I'm drawing in red bubbles proteins that, if their gene is mutated, cause recessive steroid-resistant nephrotic syndrome. And the blue bubbles are proteins from dominant nephrotic syndrome genes. So each one, gene or protein, in and of itself is necessary for podocyte function, because if you have a mutation in just one of them, there's no escaping from developing steroid-resistant nephrotic syndrome. So by finding these monogenic causes of disease, you develop a parts list that is necessary for healthy podocyte function, or that is necessary for an individual not to develop nephrotic syndrome. So that tells you all of these genes and proteins are absolutely fundamental for the disease mechanisms that are involved in nephrotic syndrome. And then if you look more closely, some of them actually have been published to interact with each other. In other words, to engage in protein-protein interaction complexes. And I drew these black boxes around them. And then for some of them, it's been published what they do. And then you can start assigning function. So not only can you find, by finding monogenic causes, the necessary components of podocyte function, but you can define these little sub-machineries. For instance, we know that slit membrane proteins are important for glomerular function, but there are also actin-binding proteins. There are actin regulators from the RORAC CDC 42 category. There are coenzyme Q10 biosynthesis proteins that, if mutated, give rise to nephrotic syndrome. And curiously, there are some excuse me, proteins or genes where we would never hypothesize that they cause nephrotic syndrome, such as many nucleoporins, which are a structural part of the holes that we have in the cell nucleus membrane, where every messenger RNA gets trafficked through. So these are fundamental genes that are highly conserved in evolution. So you would think loss of function, specifically recessive loss of function, is incompatible with life. And in fact, recessive mutations in mice are embryonic lethal, and most likely also in humans, because all the children who had mutations had one truncating mutation, but the other one was a very mild and very specific mutation in a specific part of the protein. So apparently, since these proteins have many, many functions, if you have mild specific mutations, there is a very specific disease that results from this, and in this case, this is steroid-resistant nephrotic syndrome. It's hard to believe, but there's no doubt, because we see this for 5 different nucleoporins, and the mutations actually cluster in a region. Many proteins have probably 200 plus functions in different tissues and different cell types, and nature uses them over and over for different purposes. That's probably why it's so vexing that some fundamental proteins and genes can, if mutated, lead to very specific phenotypes in specific organs. So if there are so many, I'll give you 2 examples on how we discovered some of these genes and then try to study the pathomechanisms. But before I do that, if there are 55 plus proteins and genes, we wondered what is the likelihood that we can find positive mutations in cohorts of kids and young adults who have steroid-resistant nephrotic syndrome and FSGS. And we had accrued worldwide individuals from 1,780 different families. And at the time, there were 30 genes known. We sequenced their exons in a high-throughput sequencing approach. And to our surprise, found positive mutations in 30% of them, still trying to be careful not to overcall mutations as causative. 10% were proteasome mutations, like in the child that I introduced to you initially. 7% were nephrin mutations, 5% Wnt tumor 1 mutations, 2% phospholipid C epsilon 1 mutations, and 22 other genes had to share into 6%. And this number is going up and up. There's a new gene being found every 6 to 8 weeks or so. So this high fraction of patients in which we could find monogenic causation has to be taken with a grain of salt, though, because the cohort was enriched for consanguineous families, which are more prone to have recessive disease causation, but it also, the likelihood of finding mutations was age-dependent. So the age of onset in this cohort for steroid-resistant nephrotic syndrome is plotted here on the x-axis from birth to 17 years old. And you can see that in infants, we had a 60% likelihood of finding monogenic disease causation. In toddlers, it was about 20%, and then in young adults, about 12%. And that's what we keep seeing in adults. So if there are so many monogenic causes of diseases, kidney diseases, where we didn't even expect this 10 or 15 years ago, the question is, why didn't we realize that earlier? And part of the answer is probably 10 years ago, whole exome sequencing was very much in its infancy. 5 years ago, it was still too expensive, but now it's very feasible to perform whole exome sequencing. Excuse me. But one of the other reasons is that as physicians, if we think about genetic disease causation, we usually expect familial occurrence of disease. But that's not at all true for recessive diseases, because my good friend Gregory Mendel tells me in a recessive disease, it's something like this. If you have a family has 4 kids, and the parents are heterozygous carriers, so they have one copy of a disease gene mutated, be it cystic fibrosis gene or coalescence gene. For most recessive genes, they will stay healthy. And also in the ancestry, the ancestor will be at worst heterozygous and thereby stay healthy. And then there is random segregation of gametes. So there's a 50-50 likelihood that the dad will hand on the mutation or the healthy chromosome. So 2 of them statistically have the mutation, 2 of them the healthy chromosome, same with mom. 2 of them will have the mutation, 2 of them the healthy chromosome. So statistically, in a family of 4 kids where the parents are heterozygous for a recessive disease gene mutation, you will only have 1 child who has both copies mutated and will develop the disease. You will have 2 kids who, like the parents, are healthy heterozygous carriers, and you will have 1 child that is not even a carrier. So statistically, in a recessive disease gene, you have to have 8 kids before there is familial occurrence of disease. So in other words, as a pediatric nephrologist, if you go to your clinic and there are 10 children with steroid-resistant nephrotic syndrome, you diligently take the family history in all of them, and in the 10 children, it's completely negative. That does not exclude at all that 12% or 30% of them may have steroid-resistant nephrotic syndrome due to a mutation in a monogenic recessive disease gene. So for recessive disease genes, we do not have to expect familial occurrence. Now, I would like to briefly talk about identification of novel monogenic causes of chronic kidney disease and how we do that. So here is the central dogma again: strong determinism from a gene to a disease phenotype. Over 20 years, we have accrued 10,000 blood samples and clinical data from children and young adults with chronic kidney disease worldwide, get about 2 samples per day still. We consent the patients and families that we look for the exact mutation that causes their chronic kidney disease. That's our only mandate. If we don't find it, they'll never hear from us. If we do find it and we're convinced we found the molecular cause, we're allowed, and if they want to know, they have a checkbox, then we're allowed to tell their doctor which gene and which exon. For instance, coalescence gene, exon 5, and then across the world, the doctor can talk to the patient and they can ask for what's called specific mutation confirmation. They can contact any CLIA-certified lab, because we are a research lab, we cannot generate a report. And the lab will contact us. We will tell them, for this patient, look in coalescence exon 5. They do 1 PCR for $200, not a whole exome. And then they can write a beautiful CLIA-certified clinical genetic testing report to the patient that then will be actionable. So this is a compromise on how it interfaces between research, where we cannot generate clinical testing reports, and actionable reports if the patient like to have them. So when we find one of these novel genes, we then perform cell biological studies. We make mouse models, zebrafish models, sometimes fly models, and trying to understand this black box so that at some point we will be able to target more specifically any of these disease mechanisms. I would like to give you an example for 2 brothers who had steroid-resistant nephrotic syndrome. the histology of FSGS. You see the 5 strands here, now in a different color, in red. And you can see how the podocyte foot processes, which should be nicely rhythmic, like here, where the foot processes interdigitate, that they're kind of flattened out here. That is called foot process effacement, and it's part of the phenotype of loss of function of podocyte function. The parents of the 2 boys were first-degree cousins, and that allowed us to perform something that's called homozygosity mapping, where we put polymorphic markers across the genome on the x-axis from the beginning of chromosome 1, the long arm, then we hop on to chromosome 2, short arm, long arm, all across the genome. In the 2 boys, we characterize all these markers, and these markers are polymorphic, so they can be A or B. If they're AB, that's heterozygous. That means 2 different markers. If they're A and A, that's homozygous. That means exact same marker. And if we go along the genome, we find regions, this looks like a peak, but it's actually a whole stretch of markers where all the markers are homozygous. They're all AA or BB. There's no AB. And then there were 4 other regions where this is the case. And what happens here is the parents are first-generation they're first-degree cousins. So the grandparents have a common ancestor, a common great-grandparent. And the ancestor has a chromosome that now can segregate through the father's side to the child and can segregate through the mother's side to the child. So there's a likelihood that a whole chromosome would have the exact same markers in the child because it's segregated over the father's side and the mother's side to meet as the identical chromosome from this great-grandparent in the child. However, there are crossing overs happening in every generation, recombinants. So it's never a whole chromosome that ends up to be the same in a child. The crossing overs chopping up in different smaller segments, and that's what you see here. And they're so small because we look at the combined identity for both boys. And this is actually only an 80th of the entire genome where this is the case, that the markers are identical. So unfortunately, we all have about 10 plus minus very bad heterozygous mutations in very bad disease genes. We just don't know it until we run our exome. But it doesn't matter because most recessive genes don't do us any harm if we have these heterozygous mutations, and we have one good. But if an ancestor of these boys has one of these 10, and it's a nephrosis gene, then this mutation can segregate through the dad with a, I think, 1 in 8 likelihood, and through the mom, 1 in 8 likelihood. So 1 in 64, it's a priori likely that the child is homozygous for this bad mutation. Since both kids have nephrotic syndrome, our hypothesis is there should be a nephrotic syndrome gene in an ancestor. It should have segregated and should meet as a homozygous mutation causing this disease in the 2 boys. So by definition now, we can turn this argument around. This homozygous mutation must be in one of these regions where we know the chromosomes came together identically by descent. And I'll show you why this is important in 2 slides, because we then do whole exome sequencing. That means we take the DNA from the 2 boys, we chop it up with an air jet into random pieces, and then we anneal it by base pairing to a slide or to beads that have 220,000 exons that we have in our genome. The exons are the business part of the genome. They encode for proteins, because we know every recessive disease gene mutation is 85% likelihood in an exon and not in the rest of the genome. So it's known that about 85% of all disease genes are in exons or the splice site which hook them together. So we're mostly interested in the exons because then we capture the exons by having these oligonucleotides that capture them, and that's only 1% of the genome. Then we wash out 99% of the genome because there's only a 15% likelihood that a mutation is in there. It's very hard to prove. So we just go with all the 220,000 exons. That's called the exome. Ome just means everything together. We put all this together for the 2 boys in one tube, and we do a whole exome sequencing, high throughput sequencing. And if we do this, we then align it with normal reference sequence and ask the computer for any difference between a normal reference sequence and a sequence from each boy. So if I compare my exome with yours, I'd be looking at about 4,000 differences. So that's kind of roughly the difference between people. Run the exome, you compare it between people, and you have 4,000 differences. Each one of them could be the mutation, the recessive mutation that you're chasing. So we have the problem that we have to parse down to the one that causes disease. How do we do this? There's a list of single nucleotide polymorphisms that have been published to do no harm. They're usually rare, they're less than 1%, so it should be less in the population. And that allows us to reduce twofold, because there's 2,000 of those, so we go down from 4,000 to 2,000. Then if we really, from the snippets that we caught, get rid of the introns and really only keep the exons and the splice sites, that reduces sixfold. So, but we're still looking at 325. And the hypothesis, there's one gene with one homozygous mutation. But now the whole exome, the homozygosity mapping helped us, because our sharpened hypothesis is there should be a recessive disease gene mutation which is homozygous, and it should be in one of these 5 regions where there is a risk of homozygosity by descent. These regions are only 80%, an 80th of the genome. So if we only look in these regions, we only look at 4 genetic variants. And then when my fellows inspect them, they see some of them are not real, they're from pseudogene. And so we ended up with 1. And this one was a homozygous point mutation in a gene by the name of ADCK4. And it changed in the amino acid sequence in this family an arginine in this position, 178, to a tryptophan. And I drew a little short sequence of the amino acid sequence of ADCK4 here. That's the human amino acid sequence. And you can see there's an arginine throughout evolution. So we have it, the little mouse has it, the chicken has an arginine, the frog, the fish, the sea squirt. It's a very Californian beast, that's along the coast. The fly has it, the lowly worm has it, Saccharomyces cerevisiae, that makes beer, has it, and another unicellular organism has it. So it's completely conserved throughout evolution as an arginine. So you cannot even be a happy worm if you don't have an arginine. So the patient has a tryptophan that tells you the function of the ADCK4 enzyme must be impaired. And then we went back to 1,100 other kids where we had whole exome sequencing and looked for mutations in ADCK4 and found 4 other families with homozygous highly conserved missense mutations in different parts of the protein. We also had 4 families with homozygous truncating mutations that really completely abrogate the function of the protein. So we were very confident that this is a novel gene that causes steroid-resistant nephrotic syndrome, these 2 brothers. So the question is, what does the protein do? It is part of coenzyme Q10 biosynthesis, where CoQ10 shuttles electrons from complexes 1 and 2 to complex 3. Its biosynthesis happens from mevalonic acid over multiple enzymes, and actually these 2 blue enzymes have been published to cause nephrotic syndrome if mutated, and we found mutations in CO6 and ADCK4 now to cause nephrotic syndrome. If we have mild mutations, we need to run functional tests, and in this case, we plate podocytes on a dish. We make a scratch wound in the green area. The podocytes migrate in. But if we knock down this gene, the podocytes migrate in at a slower migration rate. If I replay this, please. If we compare this to normal, they're reduced, but I'm not showing you the normal here. I'm showing you knockdown where we added CoQ10 to the cells in the dish, showing that lack of CoQ10 is part of the pathogenesis. One of our colleagues in Turkey treated one of these children where we found a mutation in CO6 with CoQ10, which is an innocuous food supplement, has no side effects. She treated her during remission, and then the parents forgot to give it, had a relapse with nephrotic syndrome, and when they reintroduced CoQ10, she responded. And there's multiple papers now that show that in this specific form of steroid-resistant nephrotic syndrome, you actually CoQ10 can mitigate the disease phenotype. So by finding the specific mutation, you realize that you can repurpose not even a drug, but even a vitamin in this disease, which is otherwise very hard to treat. So it's very important to find these children who have steroid-resistant nephrotic syndrome due to CoQ10 defects. I want to give you one more example where we found mutations in this gene that encodes RhoGDI alpha, ARHGDIA. And very briefly, it is a regulator of these three signaling proteins called Rho, RAC, and CDC42. The yellow color here shows that RhoGDI alpha labeled in red on these immunofluorescences of rat kidney sections does co-localize with all of the three because you have these yellow regions here. And in the co-immunoprecipitation, it does pull down all three of these proteins, 1, 2, 3. However, if we do co-immunoprecipitation, not with a normal sequence, but with two mutations that we found in kids with steroid-resistant nephrotic syndrome, then the protein-protein interaction is abrogated. And that tells you that pathogenesis most likely is mediated by loss of function in all three of them or one of these three signaling proteins. When we knock down the gene in a zebrafish model with a morphin renal oligonucleotide, we do this with CRISPR now, but at the time, these are happy 5-day-old zebrafish larvae. And that's the control with a scrambled sequence where no knockdown happens. But if we specifically knock down this human nephrosis gene, then the zebrafish develop periorbital edema, just like kids with nephrotic syndrome. It's a good model. And we can show that it's due to nephrotic syndrome because we do dipstick analysis in fish pee, or more specifically, we take the tank water of the little larvae and we do very highly specific ELISA. And if we do this in the control fish where there is no knockdown, they don't have proteinuria. But if we knock down RhoGDI alpha, then they develop massive proteinuria. And we can treat this with a RAC1 inhibitor, but not with a Rho inhibitor. We also saw that in the cell migration assay. Unfortunately, this drug cannot be used in humans yet, but at least it points us towards a mechanism that may be relevant for the development of new drugs to treat nephrotic syndrome. Just a brief word on cystic kidney diseases that are known as ciliopathies also, because most of their proteins localize to primary cilia and basal bodies. And all the genes that cause cystic kidneys in us or mice or zebrafish model show up at these locations of primary cilia and basal bodies. And many of these, there's 100 genes now. Many of them are syndromic, such as Bardet-Biedl syndrome, where the extrarenal manifestations are shown here, and some of these other diseases and syndromes that you know. We wanted to know in the 100 renal cystic disease genes, what is the likelihood that we can find causative mutations? And we examined 79 young individuals who had either 2 or more cysts or echogenic bilateral kidneys. And by interrogating 30 of the 100 genes at the time, we found monogenic mutations in 63% of them. And when we make the 63 100% in this pie chart, then about 64% were really cystic kidney diseases, but the others were phenocopies. They were actually all fours, renal tubulopathies, which looked echogenic and had a couple of cysts, but they were not strictly renal cystic ciliopathies. But the fascinating fact is that you can now go from a very unspecific diagnosis where a patient before the age 25 on a renal ultrasound has bilaterally chronically increased echogenic kidneys, where you usually get the report compatible with medical renal disease. Thank you very much. Not very helpful. But if you then take that as an indication to run whole exome and look at the 240 genes, you can jump in 2/3 of these individuals to monogenic disease causation. Let me, in the interest of time, let me skip over these examples. And let me just say, for chronic kidney diseases that manifest before age 25, we've talked about these leading symptoms, which are very non-invasive. It can be non-invasively accrued. So if a patient just by history has persistent proteinuria, you have anywhere between 12 and 30% likelihood of finding monogenic cause in one of these genes. If a child is known to have cystic kidney diseases, you have an almost 70% chance. In congenital anomalies, that stands at 22%. And renal stone disease, that stands at 22%. So overall, in the 230 plus genes in patients who develop chronic kidney disease before age 25, you have a 1 in 5 likelihood of finding a monogenic disease causation. And in my last slide, I would like to show you how in 104 patients who received the renal transplant at Boston Children's in the last 10 years, in the outer wheel, again, we had a similar distribution of the diagnoses and the same colors as the initial slide. Half of them had food, so on. And then in the inner wheel, we show what fraction of these diagnoses we found monogenic mutations: 18% food, 43% steroid-resistant nephrotic syndrome, 78% in cystic diseases, and so on. And this already may have consequences for clinical management, where the cocoon patients are listed here, each one in a row. Their onset or the event between birth and 25 years is shown as a triangle for first diagnosis, which mostly happens at birth, right after birth. And square is first dialysis, X is transplant, and vertical hatch in orange is when we ran the exome. So we came late to the game because it was a retrospective study. But I would say it's very important now, the minute any individual before age 25 develops chronic kidney disease, to look in these 240 genes to identify the molecular cause of disease with a 1 in 5 chance, because for each gene, there may be very different consequences. If it's Brachiotorrenal syndrome with the IR1 mutation, the patient may need a hearing test and hearing aids. If it's HNF1 beta, the patient is prone to develop MODY diabetes. So for each gene, there's very, very different clinical implications. So in my last slide, I think it's fair to say that every patient with chronic kidney disease that manifests before age 25 should now be offered molecular genetic diagnostics, because there's a 1 in 5 likelihood of finding the cause. It is the etiology, it's causative, and finding causative diagnosis may reveal personalized treatment in the future. I've shown you the rare example where CoQ10 administration will help. It helps you for your clinical studies to really know which fraction of your patients, up to 20%, has very specific diseases, so you don't compare apples and pears. It helps you to unravel the pathogenic pathways. It helps you to devise animal models, some of them high throughput, where you can develop new drugs, so that hopefully at some point we develop drugs to treat these diseases, which are very hard to treat right now. And finally, I'd like to thank the postdocs in my lab who did all the work. I would like to thank our collaborators. I would like to thank our sponsors, and I would like to thank you for your attention.
Another 5 minutes, typical nephrologist decision.
The evidence for diagnosis in your analysis?
Yeah, very good question. So do we ever see evidence for digenic mutations? In theory, it would be a mechanism, but it's extremely rare. We don't have a single case where we are convinced. We published, 3 digenic cases in renal cystic ciliopathies and nephronophthisis. Only 2 years later, find a homozygous truncating mutation in a complete novel gene, 3 different genes for all of them. So it's really rare. Until we know all the different genes, it's very hard to conclude that a digenic mechanism is happening. Just to explain, digenic means 2 recessive disease genes but the patient only has one heterozygous in one and one heterozygous in the other. That's against Gregory Mendel's rules, but you can imagine in tight protein complexes where the two proteins bind to each other and a very specific amino acid is mutated that's necessary for the binding, same happens on the other protein, then you can have digenic situation. Yes, please.
In cooking, do you think that it'll be higher yield resistance to intron because that's presumably affecting developmental processes?
Yeah, very forward-looking question because so far we were a little resigned in going to genome sequencing rather than exome because we found it doesn't contribute much. At best, we find 5% more, and then there are often copy number variants that we can find through genome sequencing. But increasingly, I think there will be a groundswell of new, very complicated mutations being found. For instance, people have described that targeted regions that are targeted by microRNAs, if they have deletions, that can give you complex phenotypes like food and others.